recognizing Ching Lau, who's really been one of my favorite interactors over the years. Every time I'm meeting Ching, we discuss a new opportunity or a new idea that he has. And he's also an Exo 1 recipient from Kids First. And importantly, we saw functional screens, germline screens. I think the next phase are additional assays that we're going to be championing as part of CBTN, and particularly as part of the translational context. Ching is at JAX Labs and really has been foundational in the germ cell working group, but also really thinking about new assay development on behalf of pediatric brain tumors. Come on up, Ching.
Thanks, Adam, for the generous introduction. This project started from a point of our research where we ran into problems, and that is we were trying to develop a project on intracranial germ cell tumors, which is a very rare type of brain tumors, mostly found in young adolescents and young adults. Because of the incidence is only about 3% of all pediatric brain tumors in the Western world. But oddly enough, it has a much higher incidence in Eastern Asian countries like in Japan, which is about 8 to 11% of all brain tumors. The other issue is we don't have a very good diagnostic marker for this tumor. So for a long time, we have been relying on two very old markers, beta-HCG and alpha-fetoprotein. Neither one was that good. People who have taken care of brain tumor patients get very frustrated when we have to rely totally on these two markers. Then the third problem was the location of these tumors are either near the pineal region or the... Oh, you can... Or you can find them in the suprasellar region. Neither one is the neurosurgeon's favorite location because it's very deep in the brain. And the risk of injuring some normal brain tissue is very high if you're trying to be very aggressive. So for a long time, we allow people to enroll patients into these trials without tissue. If one of these biomarkers seems to be consistent with the diagnosis as well as the imaging. So I have been tasked by the COG Germ Cell Tumor Committee come up with some better diagnostic assays for us. So I already told you the unmet need, and that is there are some very deep-seated brain tumors that are difficult to access. And so treatment will be initiated without these tissue diagnosis. And also, nowadays we are using more and more targeted therapy. Without these type of definitive diagnosis, it makes it questionable whether we should be using these agents. And then currently we have very little resources to monitor response to our treatment, because imaging by itself is of limited resolution. This not even impact our care of the patients, but actually the design of the whole clinical trials. I was involved in 2 generations of germ cell tumor trials at COG. We were frustrated because of the concerns that I just mentioned. So here are some of the tumors that we sometimes don't have tissue diagnosis, including DMG, as we talked about, including DIPG, hypothalamic glioma, optic pathway glioma, and then this whole group of intracranial germ cell tumors that I started out by telling you, which are mainly divided into germinoma, which accounts for about 2/3 of these type of IGCTs, and then the rest are non-germinomatous germ cell tumors. But it's a huge heterogeneous group of tumors, especially those that are of mixed germ cell tumor composition, because that is the main problem for us. If we only do a limited biopsy, that biopsy is really subjected to sampling error, depending on where the needle lands. So that was the background based on which we started developing liquid biopsy for brain tumors, initially to address this specific question. Then the next question is, should we use blood or CSF? Again, I will summarize very quickly. that our conclusion was that CSF is better for brain tumors because probably CFS is in direct contact with a lot of these tumor tissues, so you have an enriched source. And also we discovered that CSF has much less contaminant than the blood in terms of the biomarker that we are looking for. It's almost function like an ultrafiltrate. All the stuff that we don't care about, they're not there to interfere with the analysis. This is the final clincher for us to decide on using CSF instead of blood, and that is CSF harbors a lot more or higher concentration of tumor-related cell-free DNA than plasma. So currently there are a number of molecular tests that one could run with the CSF, and this includes cell-free DNA, which you could run mutational profile or single nucleotide variation. This is what most people are doing currently because probably that the most readily available. But you can also look at copy number variation or CNVs. And what we are focusing on is DNA methylation profile because ultimately that is the gold standard for molecular diagnosis of brain tumors. You can also look at total RNA, especially those tumors that have a known gene fusion. You can easily detect that if that's the case. And then you can also use microRNA. Nice thing about microRNA is it's small, probably a lot more stable even compared with cell-free DNA. And we started doing that first, okay? Here's a famous diagram that everybody has seen, underscoring why we want to do DNA methylation profiling for diagnosis of brain tumors. This was published by the Heidelberg Group in 2018, demonstrating that there are more than 100 diagnoses of brain tumors that you can readily classify by just doing DNA methylation globally. So when we try to do DNA methylation classification, we ran into some issues. One of the problems is the amount of DNA required to run the assay. Currently, if you run the tissue-based microarray DNA methylation profiling, it requires at least 200 nanogram of input DNA. Those of us who have handled CSF knows that you will never get a CSF sample of 200 nanogram unless you have like 10 ml or 20 ml of CSF. With kids, we never get consent from the parents to get that amount of CSF. This is partly because the microarray itself requires that amount of input, but it also because that assay requires bisulfite conversion, which also degrades DNA as well. So basically, we decided that we have to get away from microarray-based method and also not use bisulfite conversion. So this is what we ended up doing with the help of New England Biolab, and that is to use enzyme conversion. Their kit requires two steps. One is to protect the methylated cytosine and then cause the deamination by APOBAC. So it's a pretty straightforward assay, but it took us a while to fine-tune that for cell-free DNA in CSF. So here's the workflow that we currently use it. So we get the CSF sample from the OR, and then we spin it down. We only use the supernatant because that's where the cell-free DNA is enriched. And we use any kit that you want. We happen to use IMO kit to extract the cell-free DNA. Typically, only 1 ml of CSF has been granted. And then we use the NEB kit to do the enzyme conversion and to create the sequencing library. And then with the results, we try to classify it either using the Heidelberg classifier or the NCI classifier that Ken Aldapi group has developed. And then we compare the results. Now, because we initially tried to develop this for the diagnosis of intracranial germ cell tumors, we took advantage of the publication from Japan several years earlier, demonstrating that the difference between germinomas and non-germinomas is that there's huge global hypomethylation in the germinoma cases. As indicated here, the blue samples are the germinomas, the red ones are the non-germinomas. And you can also see that the amount of methylation globally is much lower among the germinomas. So we said, oh, that's good. That's an easy shortcut for us. We just look for global methylation profile and see if we can diagnose germinoma from non-germinoma. So here's a summary of the first few cases that we did. It's color-coded. The first 5 cases are pink. They are non-germinomas. And then the bottom of the panel, they are blue, and they are germinomas. As you could see, the level of methylation as plotted on the y-axis goes from 0 to 1, 1 being fully methylated, 0 being fully hypomethylated, and the results are plotted according to the chromosomal locations. You can see that the germinoma cases, overall, they are very low methylation status, typically less than 20%, versus the non-germinoma cases on the top, they are much higher. So that confirmed what the Japanese colleagues have published, and that was very helpful for us. But we still wanted to try to this is a composite of those cases. Even when you combine all the cases, you can easily separate the germinomas from the non-germinomas. And throughout these type of analysis, we began to learn how we could get the assay to run better, and that is to monitor the amount of cell-free DNA that we can extract from that 1 ml of CSF. And so for us, the cell-free DNA are usually found between 120 to 200 base pair. And you can see there are different scenarios where we could have very clean cell-free DNA with no contamination from the cellular DNA. And it turns out that if the profile of the cell-free DNA is clean and the amount is adequate, we can almost guarantee we will get the classification done. So this is actually not just looking at the global methylation profile, but actually a formal classification of these tumors. As you could see with the germinomas, we never had problem, partly because the cell-free DNA peak is very clean, and the prediction score is very reliable, versus the non-germinomas always have problem if this is a mature teratoma. I think people know what is going on if you know the biology. And also you notice that if it has trouble classifying, it called ependymal family. Brian and I were joking about this. This is probably from the chloroplexus, because that's where the CSF is generated. So we still have some work to do with the non-germinoma germ cell tumor. But fortunately, as I said, just using the global methylation profile, we can still reliably separate them from the germinoma. Then I just want to spend the last 30 seconds to tell you that we now have broadened this to study non-germ cell tumor, including medulloblastoma and high-grade gliomas. And again, they follow that same rule. If the cell-free DNA peak looks good by tape station profiling, we can almost always guarantee that we can classify these. So we started titrating the amount of input DNA using these other cases as low as 0.1 nanogram, and also directly comparing the profiles that are generated from CSF versus blood, and also comparing sequencing versus microarray. As you could see, this is one case, a medulloblastoma case. We can adequately classify down to the sub-class level with very reliable prediction score, versus the tissue and the microarray. As far as tissue is concerned, it matches that of the CSF completely. But the plasma of the same patient collected at the same time as the CSF, we couldn't adequately classify that. And then when we use the array-based method for methylation, again, CSF-based sequencing method comes out ahead. And this is the same again using another case of high-grade glioma, very accurate classification based on methylation. And in addition, we can also pick up all the copy number variation, at least at the chromosomal levels. As you could see, we compare that with the tissue cytogenetic profile. It matches that beautifully. Here's another case with medulloblastoma. Not only that we can pick up the chromosomal arm level of CMV, but we can also pick up these very focal copy number changes. Here's a case with TP53. Notice that we, in this case, we use 10 nanogram of input, but we can also pick up these abnormalities at much lower concentration. This is a CDK N2A copy number change. To our surprise, when we went back and looked at the CMVs of germ cell tumors, germinoma is highly, highly unstable genome, as you could see here. Almost every chromosome arm has alteration. Versus the teratoma, the mature teratoma, very stable. Again, this is using only 1 nanogram of input DNA. So not only that we could potentially make the diagnosis without any tissue diagnosis in a situation where the location of the tumor is too delicate, but we could also consider monitoring the response to treatment now because of these detection of copy number changes. And potentially we can work like the leukemia folks to start looking at the MRD status type of evaluations and predict prognosis. And hopefully can also pick up relapse or progression early on. So these are the conclusions that I told you. I think there's potential for this assay. So I'm looking to CVTN to continue our collaboration to expand this to other type of brain tumors. And again, this takes a village to do, and these are the people that are involved in this whole project, including the funders. And so I thank you for your attention.
So before Adam takes you away, a few questions. Have you looked at the different sources of CSF collection and if there's variation between if this was a lumbar puncture versus EVD? Does it affect your methylation or the amount of cell-free DNA that you get?
Yes. Can you hear me? Yeah. With the limited number of cases that we have, we started looking at that possibility that, for example, we had some samples that were actually collected from the cisternal fluid. Jay would know that when you open up the skull, a lot of surgeons like to decompress the brain by taking some of the cisternal fluid out first. In the past, they just discard that, and we requested that they save that and compare that with the CSF, really compare with a lumbar puncture. There is some slight difference. Okay, and also whether you collect it from an EVD or not. So eventually we have to standardize the collection of the CSF in order to properly interpret the results. Yeah.
And then for methylation, you also have when you have a nice peak of your cell-free DNA, you also have DNA that was contributed by healthy cells.
Yes.
How does that confound your methylation data?
Thank you for asking. I didn't have time to go over that. Remember that the cell-free DNA are small, less than 200 base pairs, right? The way we construct the sequencing libraries, we do not fragment the DNA. So the potential contribution coming from the cellular DNA contamination would be minimized because it favors the smaller molecular weight DNA. And I think that may have contributed to some very clean results that we have, even when there is another contamination peak that is of much higher molecular weight.
what Ching is sort of highlighting, right, that CBTN's focus on sample collection and data generation, right, can be expanded now into that longitudinal context and other sources. And Ching already has a project within CBTN to advance it.